So today I'm going to be presenting SC Performer, a semi-supervised transformer-based model for single cell type classification. So to start off, single cell type classification is an important first step for many downstream analyses in a single cell world. And manual single cell type classification is typically done by taking a cell by gene expression matrix, which we can derive from a single cell RNA sequencing experiment. We can then cluster those cells based on the similarities between their gene expression values, and then identify a set of marker genes characterizing each cluster, and label cells accordingly based on domain expert knowledge. Now, even though this is extremely important to develop ground truth cell type labels, it has many drawbacks. It's laborious and costly and doesn't scale well to large data sets, especially since single cell RNA sequencing data sets are going to millions of cells now. And in addition, it's at the mercy of whatever clustering method you choose, so it's subject to bias as well. And so one solution to overcoming this is to use machine learning to assist researchers in classifying these single cells. Now, the way machine learning models work in single cell type classification is they're first trained on a reference data set with known cell type labels. And using that trained model, you can then perform single cell type classification on a target data set. And so before I dive into SC Performer, I want to introduce three different machine learning paradigms and methods. So the first is transformer. I know this has been mentioned before, but I just want to briefly introduce it. So transformer is a deep learning architecture that was originally applied in the natural language processing field. And one of the domains of application is translation. And so as you can see here, you feed it a sentence in one language, it spits out the same sentence in another language. And at a very high level, transformers are nothing but an encoder and a decoder. And I'm going to focus on the encoder architecture here, which is will be most useful for our case. And so the encoder part of the transformer essentially takes these vector representations of each word in the sentence. And you have this self-attention layer, which essentially computes all pairwise interactions between the words in the sentence. And so by doing so, you're able to learn these long-range interactions between all words in the sentence, even if it's syntactically very complex and even if the sentence is extremely long. And in the end, you spit out these new vectors, representations of each word, which contain information from all other words in the sentence. Then you feed it through a feed-forward neural network, which learns even more complex representations or complex interactions. And you, in the end, get same dimensional vector representation of each word from this encoder architecture. And with that output, you can then perform all kinds of downstream analyses. And so to bring it back to single cell biology, we can apply transformers to the single cell domain by thinking of cells as sentences and genes or groups of genes as words. And so the idea here is that with a transformer architecture, you might be able to learn more complex gene-gene interactions that could capture these cell type definitions. And so that was our hope in creating this model. And the second machine learning paradigm I want to introduce is semi-supervision. So supervision, supervised learning is the classic case in which all of your training data is labeled. And semi-supervision, in contrast, only some of the training data is labeled. And this is particularly well suited to single-cell RNA sequencing data because most of the data out there is unlabeled and it's constantly being generated. Data sets are becoming larger and larger. So a machine learning model that exploits this is going to be more able to learn these general representations of each cell type. And then the last machine learning paradigm I want to introduce is a domain adversary, which is a model that performs a binary prediction by assigning a label for each cell and saying whether it's part of the training data set or the test or the target data set. And what this does is you can stick such a model into whatever architecture you have, and it makes your model more generalizable, and it's better able to learn domain invariant representations of your data. And so with these three paradigms in mind, We developed SC Performer. This is an overview of the architecture of our model. And so it takes the full transcriptome as input, performs a dimensionality reduction using a fully connected network. We feed this low dimensional vector representation into a transformer. Part of that transformer's output is then fed into this cell type classification head, which is what's actually performing the cell type classification. Then the rest of the transformer's output is fed into this reconstruction head, which is essentially predicting the gene expression values of genes that were masked during training. And then finally, we have this domain adversarial head, which is what I mentioned being how the model learns to distinguish between training and target samples. And so this classification loss, imputation loss, and adversarial loss are all minimized in parallel. And that is how SC Performer learns the cell type classification. And so we use two methods of evaluation, intra-dataset and cross-dataset. So in the intra-dataset case, what we do is we split the dataset into a training and testing domains. And we do this by splitting according to different domains. These could be different patients, diseases, technology. So we might train on, let's say, samples from old rats and then predict on samples from younger rats. And then finally, we also use the cross dataset strategy, which is training on one dataset and testing on another. And by doing that, we can test the model's ability to learn domain shifts according to different labs conducting different experiments. And so these were the datasets we used for the intra-dataset analysis. And as you can see, they vary significantly in terms of the number of cells, tissue of origin, number of cell types, and how we split across different domains, or what domains we split the data across. So age, sequencing technology, different donors, experimental batches. And so we computed accuracy across three models. So SC Performer, SC9, which is a state-of-the-art neural network model that also employs a domain adversary and semi-supervision, developed by David Kelly and Jacob Kimmel, and a baseline logistic regression model. And as you can see, SC Performer is overall the best performing model, although only marginally so. Next weekend, we looked at the mistakes that SC Performer is making. And so this is in the T-cell data set, and hope it's visible. As you can see, these two confusion matrices, cells whose ground truth label is TH star, a sizable proportion of them tend to be predicted as either TH1 or TH17. And TH star is a heterogeneous cell type that is defined as having characteristics from both TH1 and TH17 cells. And what's interesting that we found is that cells whose ground truth label is TH star but was predicted as TH1 or TH17 tend to be transcriptionally similar to TH1 and TH17 cells respectively. And that's what the figure on the right is showing across 8 marker genes. And so the idea here is that even though SC Performer is incorrectly labeling these cells, they could indeed be TH1 or TH17 cells, and that SC Performer is indeed performing the correct classification. We can also see from these confusion matrices that naive and memory Treg cells are also tend to be confused for each other, and by both models, both SC9 and SC Performer. And this makes sense because they have shared gene expression profiles, and they both express FOXP3. Now, in terms of interpretability, we can look at SC Performer's cell embeddings and whether they can better separate cell types, this UMAP space. And so on the left, you have a UMAP derived from the original gene expression values in the cortex dataset. And as you can see, there is some overlap between the 5 different neuronal cell types, particularly the excitatory and inhibitory neurons, which should be quite distinct in this space. But if we use SC Performer's cell embeddings, we can see much better separation between the 5 different cell types. And these embeddings are the output of the transformer module in our model, and it's what we feed into the classification head. Moving on to the cross dataset comparisons, we decided to include two additional data sets, which I term Hanifa and Zumin. These are much larger than the intra data sets, and they comprise 18 different coarsely defined cell types, and they are COVID cell atlas data sets. And so the idea, again, here is to train on one data set and test on another. And before we can compute accuracy in this case, cell type labels are going to differ, right, based on different labs, different experiments. And what we found is that most major cells have a one-to-one mapping between Hanifa, Zumin, and the Tsang data sets. And we decided to compute accuracy in this way that if a model predicts a subset or superset cell type, it is counted as a success. And this little schematic here is what I mean by superset and subset cell types, because cell type definitions are very much hierarchical in structure. And cell types not found in one data set or not found in the other data set are left as is. And so using this approach, we can then compute accuracy. SC9 does perform better in this case, although SC performer is still competitive for most cases. And here I'm just looking at all the different permutations by training on one data set and testing on another. And now moving on to what I call fine cell type analysis, you can ask the question, how does the distribution of predicted fine cell types, these are very specific subcell types, across different disease states in the test data set, and compare that to the distribution of true fine cell types across the same disease states, but in the training data set. And so a model should be able to capture these distribution changes in the target data set. And that is indeed the case. And you can see these distributional changes that are captured by SC Performer and SC9 by training on Zumin and predicting on Hanifa. And so with that, I want to conclude that SC Performer overall is competitive and comparable to SC9 on intra-data set tests. SC Performer cell embeddings better separate cell types when a domain shift is present in the data. It performs worse than SC9 overall in cross-data set tasks. And finally, SC Performer is capable of finding these fine cell type predictions across disease states. So with that, I want to thank Abbas, Ferhadai, and the rest of the members of the iLab, as well as the ANH T32 training grant for funding my research. Thank you all for listening.
Questions? Sure.
Appreciate you finding our method. I'm curious, I don't think I quite followed the use of the transformer. So the self-attention operation is happening over multiple cells which have been embedded by the neural net? Is that right? Or
it's multiple genes, essentially.
So the genes have a channel vector at that point?
Essentially, yeah.
And where does that come from?
That's learned by another neural network, and everything is trained jointly. So that's how. Because originally the model was used for like just a set of highly variable genes, but we decided to move to a full transcriptome, and so we figured that we would, because single-cell RNA-seq is of course very noisy, we would perform this dimensionality reduction step as part of the training process. So that's how we get these gene vectors.
And is the gene vector based on its expression across all the cell types? Is it kind of like transposing the gene expression?
Yes,
vector?
it's across all of the cell types, yeah.
Okay.
Yeah.
Do you have any anecdotes about the value of the transformer from your analyses?
Any anecdotes? What do you mean exactly?
Like, what is the transformer? How is it modifying the representations in a way that's useful?
Okay, yeah, we have not looked directly at that, but that's an interesting suggestion, yeah, to see like how that particular architecture would help with that. Yeah, thanks.